Hello students, welcome to Simplify Pharma. Do subscribe to my channel to stay updated with the latest videos. Today we are gonna study about Remdesivir. What is Remdesivir and what role it plays in Covid-19 situation across the globe? Being a pharmacist, it is very much essential to know about Remdesivir. This is the chemical structure of Remdesivir. It is a broad spectrum antiviral agent. It is a prodrug of GS441524, that is adenosine. So for that we need to understand what do you mean by a prodrug. Prodrug is an inactive form of a medication. After administration, it is metabolized, that is converted into a pharmacologically active drug within the body. This is a simple example of prodrug and active drug. In prodrug, once the drug reaches inside the body, it gets metabolized. That is, the drug becomes active only after metabolism. Whereas in active drug, the moment the drug is inside the body, it is already active and it starts showing its effect. So similar manner, Remdesivir is also a prodrug. That means it will be inactive form. It will be in inactive form once it is outside the body, but once it is inside the body, it gets metabolized and then it is converted into active form. So Remdesivir is a broad spectrum antiviral agent. It is a prodrug of GS441524, that is adenosine. So this GS441524 is the active form of Remdesivir. And this adenosine, it is used by virus to produce more of its viral RNA. Remdesivir is a nucleotide analog. That means it mimics adenosine, one of the building blocks of RNA viruses genome. Now when we say analog, it means a mimic. It is not a real thing. Though it looks like the real thing, but it's not the real thing. Means it is adenosine analog. It looks like adenosine, but in true sense, it is not adenosine. It is a prodrug of an adenosine triphosphate analog with potential antiviral activity against a variety of RNA viruses. So it is a nucleotide analog prodrug that perturbs viral replication by inhibiting RNA synthesis. It was developed by biopharmaceutical company Gilead Sciences in the year 2009 as part of the company's research and development program for hepatitis C. Though it did not work against hepatitis C, but was then repurposed and studied as a potential treatment for Ebola virus disease and Marburg virus infection. Now, when we say repurposed, what does a repurposed drug means? Drug repurposing, also called as drug repositioning, reprofiling, or retasking, it is a strategy for identifying new uses for already approved or investigational drugs that are outside the scope of the original medical indication. Now, this strategy offers various advantages over developing an entirely new drug for a given indication. It is cost-effective, rapid alternative to identifying new drugs. Drugs have already met with FDA approval, allowing the early stages of drug development to be bypassed. By mining repurposed drug libraries for new indications, researchers can rapidly move to the clinical testing portion of the drug discovery process. So this approach dramatically shortens the development timeline and gets new treatments to patients faster. So this drug is already there in the market. It is known for a particular disease, just that we are exploring it for altogether a new disease. Now, since this drug is already there in the market, that means it has already bypassed all the stages of preclinical trials, clinical trials. So it is easier to get approval for this drug for a new indication. This is called as repurposed drug. Now, Remdesivir is an investigational nucleotide analog with broad spectrum antiviral activity. Now, what do you mean by investigational? It means it is not approved anywhere globally for any use. It is still not approved. Dosage is initial one-time dose of 200 milligram followed by 100 milligram per day to complete a 10-day or a 5-day course. Route of administration is intravenous, parenteral. So you can see in this diagram, this is the prodrug Remdesivir. Outside the body it is inactive. The moment it reaches inside the body, it gets converted into active molecule that is called as GS441524. Elimination half-life. The nucleoside triphosphate metabolite has a half-life of approximately 20 hours in humans. Side effects. Common side effects include nausea, vomiting, and serious side effects, they include increased liver enzyme levels that may or may not indicate liver damage. That is, we can see an elevation in levels of transaminase. It is a type of liver enzyme. Now let's come to the mechanism of action of Remdesivir. Now, we all are aware of this. Coronaviruses, they have a single stranded RNA as their genetic material. RNA viruses are dependent on RNA polymerase enzyme to grow the RNA chain. So when the novel coronavirus SARS-CoV-2 enters a human cell, an enzyme called RDRP helps the virus to replicate. We are going to discuss this mechanism in detail in the further slides. So drugs that act as nucleotide analogs interfere with the RNA-dependent RNA polymerase, that is RDRP, enzyme that the virus uses to copy their genomes. So what exactly is Remdesivir doing? It is working by inhibiting the activity of RDRP. Remdesivir is inhibiting the activity of RNA-dependent RNA polymerase enzyme that the viruses use to replicate. So this drug Remdesivir is designed to obstruct the novel coronavirus at the stage of replication. Now, upon administration, Remdesivir, being a prodrug, is metabolized into its active form, GS441524. It is an ATP analog that competes with ATP for incorporation into RNA and inhibits the action of viral RNA-dependent RNA polymerase. This results in the termination of RNA transcription and decreases the viral RNA production. Remdesivir has demonstrated in vitro and in vivo activity in animal models against the viral pathogens MERS and SARS, which are also coronaviruses and are structurally similar to COVID-19. The limited preclinical data on Remdesivir in MERS and SARS indicate that Remdesivir may have potential activity against the COVID-19. This was as mentioned on the site of GLEAD. Now, Remdesivir is an experimental medicine that does not have established safety or efficacy for the treatment of any condition. So, what was the GLEAD's response to COVID-19? It entails three main areas: Remdesivir clinical trials, emergency access to Remdesivir outside of clinical trial, and increasing the manufacturing and supply for Remdesivir. So, this is one paper that we can access. Remdesivir and chloroquine effectively inhibit the recently emerged novel coronavirus in vitro. Now, in vitro means it is in cell culture dish, in petri dish. only in certain types of cells. It did not show results in human. The clinical trials are still going on. There is one more paper. Coronavirus susceptibility to the antiviral remdesivir is mediated by the viral polymerase and the proofreading exoribonuclease. Now, before we move further, in the previous paper, I would like to explain this GS5734 is another name for remdesivir. So before we move further and understand in detail the mechanism of action of remdesivir, we need to understand few terms like endocytosis. It is a cellular process in which substances are brought into the cell. The material to be internalized is surrounded by an area of cell membrane, which then buds off inside the cell to form a vesicle containing the ingested material. There are different types of endocytosis, like phagocytosis, in which the solid particle is taken inside the plasma membrane by forming a phagosome. There's something called as pinocytosis, in which the extracellular fluid, it enters inside forming a vesicle. Then there's something called as receptor mediated endocytosis, where the different proteins, it comes and attach itself to the receptors that are present on the surface of the cell, and by attaching to these receptors, the proteins are able to enter inside the cell by forming a coated vesicle. And we also need to understand the different components of a typical animal cell. This we all have studied. Now in this diagram, the two components that we need to understand and their function we should know before going any further is ribosome. If you can see the little dots, that is the ribosome. And on this endoplasmic reticulum, the small dots that you can see, this is a ribosome. And what's the function? It's a site of protein synthesis. Then coming to lysosome, you can see over here, and the function to break down unwanted macromolecules. So this is a simple animation wherein you can see how ribosome is involved in protein synthesis. Okay, now let's come to our mechanism of action. In this diagram, we can see this is the host cell membrane of the host cell, the receptor that is there on the cell membrane, surface of the cell membrane. This is the lysosome. This is the nucleus of the host cell. This is the COVID-19 virus that has spike S proteins on its surface. So the lysosome is responsible for cycling of waste products. Nucleus, it contains the genetic code. The virus, it contains spike S proteins on its surface that helps it to bind to ACE2 receptors. So with the help of the spike S protein, the virus comes and bind to this ACE2 receptor on the surface of host cell. So once the virus binds to this ACE2 receptor on the surface of host cell, it enters the cell through endocytosis and forming an endosome. So we discussed earlier endocytosis, taking in of matter by a living cell by invagination of its membrane to form a vacuole. So the next step would be this endosome will transverse through the cytosome and try to fuse with lysosome. This endosome will try to fuse with lysosome. Then after that, what happens? The coronavirus is released out of endosome or lysosome and it spreads out. The COVID-19 virus is a single-stranded RNA virus. It's a single-stranded RNA virus. So this viral RNA is the genetic code of coronavirus. The way our DNA is made up of nucleotides, viral RNA is also made up of nucleotides. There are 4 nucleotide bases in viral RNA, little different from DNA. So you can see over here, these are the 4 nucleotide bases of viral RNA: adenine, guanine, cytosine, and uracil. Whereas these are the 4 nucleotide bases of our DNA: adenine, thymine, guanine, and cytosine. So you can see this, the viral RNA doesn't have thymine, it has uracil instead of a thymine. So what next happens? Once the virus it enters the human cell, what it does is it releases its genetic material, which is then copied using the body's existing mechanism. It takes over the host cell's machinery, like the ribosome. Ribosome's main function is to synthesize protein. So this way it releases the genetic material which is copied by ribosome, and after that we can see the ribosome look at this piece of viral RNA and translate it into RDRP, which is also a viral protein. So what is RDRP? RNA dependent RNA polymerase, RDRP. It is also known as RNA replicase. It is an enzyme that catalyzes the replication of RNA from an RNA template. Now this is obviously in contrast to typical DNA dependent RNA polymerase, which catalyzes the transcription of RNA from a DNA template. So at the replication stage, the key protein of the virus at play is this enzyme, RDRP. Why? Because RDRP makes copies by processing components of the RNA of the virus. So scientifically, such an enzyme is called polymerase or replicase. It catalyzes the synthesis of the RNA strand complementary to the given RNA template. So as the name suggests, RNA dependent RNA polymerase, it is dependent on RNA, so it needs RNA to function, and RNA polymerase means it makes RNA. So it produces multiple copies of viral RNA. So these viral RNA can produce more proteins like the spike S protein, and thus the virus leaves the cell. You can see multiple copies of virus are produced, replicated, and they leave the cell. So this happens in each and every cell, and thus millions and millions of this virus are replicated and they attack the host cell. So what happens when the patient takes remdesivir? The remdesivir, it enters the cell and gets converted into adenosine analog. It means it looks and acts like adenosine, but that's not true. Now GS441524 is an adenosine analog. It's a nucleoside. It acts like, but is not same as a nucleotide base on RNA. A nucleoside is a nucleotide without a phosphate group. So though it mimics the A nucleotide base of RNA, but it is not in actual. So this RDRP gets confused between GS441524 and A base. And in this confusion, it uses GS441524 for making viral RNA. So here it does a mistake by incorporating GS441524 instead of A base in making viral RNA. So it results in defective or faulty viral RNA production, and this defect, it stops RDRP from replicating further viral RNA. So there are again few research papers. Coronavirus susceptibility to antiviral remdesivir is mediated by viral polymerase and the proofreading exoribonuclease. Now we'll see what is this exoribonuclease. This mechanism we studied, the remdesivir, it enters this host cell. It gets converted into GS441524, that's a prodrug, and it mimics one of the nucleoside. And this is the reason RDRP uses this instead of that nucleotide, and it incorporates that faulty nucleoside, that is GS441524, in this viral RNA. While replication, it results in formation of faulty viral RNA, and that stops the replication of viral RNA any further. Now this virus is very smart. Many a times it happens, this COVID, it has proofreading non-structural protein that is called as NSP14 3' to 5' exoribonuclease, and that is required for normal COVID recombination, and that its genetic inactivation causes significantly decreased frequency and altered patterns of recombination in both infected cells and released variants. So in simple language, there is something called as 3' to 5' exoribonuclease. This is also produced from original viral RNA strand. So that means it is a viral RNA protein. And what is its function? Its function is to detect and fix problems and defects in viral RNA strand. So it can remove any faulty base to help the RDRP produce normal viral RNA. So exonuclease acts contradictory to effects of remdesivir. This exoribonuclease has been shown to be part of viruses like coronavirus. So as I said, coronavirus is a very smart virus. Even though we are giving it remdesivir so that we can block the replication of the virus. How? Because remdesivir mimics one of the nucleotide, and by mistake the RDRP uses it to produce faulty RNA. So in that case, what does our smart virus does? It produces one more viral protein that is called as exoribonuclease. And what is the function of exoribonuclease? Its function is called as proofreading, that is to detect and fix problems and defects in any viral RNA strand. And if it detects the fault in this viral RNA strand, it will fix it. It can remove that faulty base to help RDRP to again start producing normal viral RNA. So the replication of viral RNA again will start. So remdesivir has shown antiviral effects in vitro studies in human respiratory epithelial cells against SARS-CoV and MERS-CoV. Remdesivir has shown positive effects in vivo animal models. There is a paper for that. You can refer it for further details. Remdesivir, again, a review of its discovery and development leading to emergency use authorization for treatment of COVID-19. Coronaviruses lacking exoribonuclease activity are susceptible to lethal mutagenesis, evidence for proofreading and potential therapeutics. So what they have mentioned in this paper, many a times it happens. In the previous paper they have mentioned, as you increase the concentration of remdesivir, the viral RNA replication will decrease. But many a times it happens, there is a mutation in this exoribonuclease, and cause of that mutation, what will happen? This remdesivir will no longer show its action on this COVID-19, and COVID-19 will develop resistance to remdesivir. So these are the different references. I really hope you liked my video. That was a simple effort in order to simplify the mechanism of action of remdesivir, to understand what is remdesivir, why is it so much in demand, why is it so much in news, why is it so much correlated with COVID-19. And being a pharmacist, we should be aware about remdesivir. Do subscribe to my channel and do leave in the comment box if you like the video, if you have any doubts regarding remdesivir. Thank you.